Alright, so today I'm going to be talking about the structure of RNA versus that of DNA. RNA and DNA both have nucleotides, but there are some differences that we will see. For example, RNA contains ribose, which has an hydroxide group on the 2' end, and DNA has only a hydrogen on the 2' end. They both have bases, but RNA's base contains uracil, whereas DNA's base contains thymine. So both of them has phosphate groups attached to the 3' end as well as the 5' end, which creates a phosphodiester bond between the sugar and the phosphate group. So I would also like to note that DNA is a double helix. It's double-stranded, whereas RNA is single-stranded. So because RNA is single-stranded, it's able to create this hairpin structure with complementary nucleotides. Which is cool. Now that we know what an RNA molecule looks like, we should now look at the process of transcription. Transcription is essentially the synthesis of an RNA molecule from the DNA template strand. So therefore it happens before translation of RNA to protein. It's very similar to replication, except that only certain parts of the RNA are transcribed. Now, we need a DNA template strand as well as some raw materials and a transcription apparatus to get the transcription process going. So, let's open up our DNA molecule to see how this works. So, the RNA molecule will come in and it will synthesize from the 5' end to the 3' end because it is anti-parallel and complementary. Keep in mind that RNA contains uracil and DNA contains thymine. The non-template strand is not synthesized or doesn't play a role, but the bottom strand is the template strand that is needed. So, in most organisms, each gene is transcribed from a single DNA strand, but different genes can be transcribed from either DNA strand. So some RNA will bind for the A gene, and some will be for the B gene, as well as the C gene. Either template strand can be used. So, check DNA template strand. Something else to note is that the transcription unit is very important in the process. It includes a promoter, which is where everything begins. It includes the RNA coding region, where RNA will be encoded, and also the termination site, where all of this will come to an end. Again, the RNA is synthesized from the 5' end to the 3' end. So, next we need to look at the substrates, or the raw materials that are needed. And basically the only one we really need is ribonucleotide triphosphates. Basically has a sugar, a base, as well as a triphosphate molecule attached to the 5' end. Now let's look at the apparatus. The apparatus are proteins that have 2 beta molecules, 2 alpha molecules, and a omega molecule. there's something called the sigma factor that binds to it in order for the whole transcription process to take place, which binds to the promoter. Like this. Now let's look at bacterial transcription. So bacterial transcription involves initiation as well as elongation and lastly termination. Initiation itself includes the promoter recognition, the expansion of the transcription bubble, and the creation of bonds between the R and TPs, as well as the removal of the transcription apparatus. So the bacterial transcription has a Tata box, and the Tata box is on the promoter. It's actually a consensus sequence, which is basically just a short stretch of common nucleotides where the RNA polymerase can recognize that region and bind to it. Once the RNA polymerase binds to it, the sigma protein also binds to it, and together they are creating the holoenzyme, which binds tightly and unwinds the double-stranded DNA in order to expand the bubble. Keep in mind that anything before the start of transcription relatively is upstream, and after that is downstream. So let's look at how the DNA molecule is expanded and unwinded. Once the bubble opens up and the RNA polymerase and the sigma bond are on there, the RNA strand can come in and bind to the region. Nucleotides are added to the 3' end of the RNA. Now there is 3 phosphate bonds on one of the nucleotides, but 2 of them fall off and it just creates 1 phosphate that is left. However, at the 5' end, all 3 of the phosphate remain on the RNA. Once the sigma bond of the RNA is released, polymerase moves beyond the promoter and continues its journey of transcription. Elongation follows the process of initiation. It's actually a pretty short process that just creates the transcription bubbles, which is typically about 18 nucleotides long. Positive supercoiling occurs ahead of the transcription bubble, while negative coiling occurs behind the transcription bubble. 2 things that happen during elongation include backtracking as well as proofreading. There's actually a lot of backtracking and pauses that happen, especially if there's a lot of secondary structures, but ribosomes come in and try to lessen the amount of backtracking that occurs. The enzyme called topoisomerase helps in the process of coiling and supercoiling and all of that jazz. So that's basically it for elongation. In the process of termination, the RNA doesn't stop right when it gets to that sequence, but rather it transcribes that sequence and then it stops. It's like a car going after, stopping like after it hits a speed bump. So the first type of transcription termination we're going to talk about is the row-dependent one. So the row binds to the rut site and it moves towards the 3' end. So when the RNA polymerase encounters a terminator sequence, it pauses. And when it pauses, it creates this hairpin structure. And then the row protein will get a chance to catch up. Using helicase activity, row unwinds the RNA and DNA hybrid, and it brings an end to transcription. The other type of termination is called the row-independent termination process, and that just basically means it doesn't need the row protein to stop. The row-independent terminator contains an inverted repeat followed by a string of approximately 6 adenine nucleotides, which is not drawn here, but just imagine. So this inverted sequence repeat is transcribed onto the RNA, and that just basically creates the hairpin structure on the RNA strand. The hairpin strand actually destabilizes the RNA and DNA pairing so that the two can actually separate from each other at the end of transcription. I wish I had drawn this, but basically the string of U's causes the RNA polymerase to pause, and then the RNA transcript separates from the template, ending the process of termination. Eukaryotic transcription is a little bit different from bacterial transcription because it starts out as a histone, but then an acetyl transferase comes in and adds the acetyl groups in order to destabilize all of it in order for the process of transcription to actually begin. As for promoters, eukaryotic transcription requires the regulatory promoter as well as the core promoter. The core promoter is downstream from the regulatory promoter, and it has some sites that encode certain sequences, but the most important one is the TATA box, where the polymerase can bind to. The regulatory promoter is basically just responsible for the rate of transcription. It's basically where the activator protein binds to, as well as the enhancer. Now, there are certain other proteins, such as the mediator, and complex, like general transcription factors that stand for certain things. These proteins basically replace the sigma factor that is seen in bacterial transcription. Keep in mind that the RNA polymerase 1 and 3 recognizes distinct promoters. But anyways, the TFD binds to the TATA box in the core promoter, along with the TBP. The TBP is also known as the TATA binding protein. Then the transcription factors in the RNA polymerase 2 also binds to the core promoter. The DNA loops out, allowing the proteins bound to the enhancer to interact with the basal transcription apparatus. The transcription activator proteins bind to the sequence in the enhancers. And the TAPs, transcriptional activator proteins, bind to the sequences in the regulatory promoter and interact with the basal transcription apparatus through the mediator. Initiation is just when the TF2 binds to unwind and to make a template to form an open complex as phosphate groups are cleaved and nucleotides joined together. Now let's talk about elongation and termination. The DNA double helix enters a cleft in the polymerase and is gripped by jaw-like extensions of enzymes. The two strands of DNA are unwound, and RNA nucleotides that are complementary to the template strand are added to the 3' end of the growing RNA molecule. When it funnels through the polymerase, the RNA and DNA hybrid hits a wall of amino acids that binds and makes a right angle. The newly synthesized RNA is then separated from the DNA and it runs through another groove before exiting the polymerase. Now, in termination of eukaryotic transcription, there's not actually any hairpin secondary structures. There's actually a protein called the RAT1, which is very similar to the Rho protein factor. So the RNA2 polymerase transcribes well past the coding sequence of most genes, and the RNA is cleaved near the 3' end of the RNA. The RAT1 endonuclease actually attaches to the 5' end of the trailing RNA sequence, and the sequence that is cut off is called the pre-mRNA. But the RAT1 starts moving toward the RNA polymerase, degrading the RNA as it goes, just as a rat would eat some cheese or something like that. And once the RAT1 reaches the polymerase, transcription is terminated, and the RNA polymerase 2 comes off. And then the RNA separates from the DNA to end the process of transcription in eukaryotes.